Hello everyone, this is Pep and this is Shu and this is me, Zal, and next to me is Fizri. Our group would like to present about spectrophotometry, protein analysis using Bradford methods. So we have three objectives. The first one being to determine the absorbance of protein at different concentration using spectrophotometry. Next is to construct a calibration standard curve for protein concentration. And the last one being to estimate the concentration of unknown protein samples of A, B, and C. Spectrophotometry works based on the Beers-Lambert law, where absorbance is directly proportional to concentration.
For the materials, firstly we have distilled water. It is a solvent used to prepare buffers and act as a blank to zero the spectrophotometer when using protein samples. Next, we have 70% ethanol. It is used in the lab to sterilize and clean surfaces and equipment to prevent cross-contamination. Next, we have Bradford reagent. It is a specific chemical reagent that is used to react with proteins and produce a colored product. They can be measured at 595 nanometer. Then you have test tubes and test tube rack. It is used to hold and mix the chemical reactions and hook, while the rack provides a stable base to hold them upright. Furthermore, there are three unknown protein solutions, which is A, B, and C, that contains unidentified concentrations that must be determined by comparing their absorbance to the BSA standard curve. Next, we have a cuvette, which is an optically transparent cells designed to hold the liquid samples during measurement. Next, we have a spectrophotometer, which is an instrument used to measure the intensity of light absorbed by the sample or the intensity of light transmitted through it. Then we have a micropipette. It is an adjustable mechanical device used to accurately measure and transfer very small volumes of liquid, ranging from 5 microliter to 1000 microliter. And lastly, we have a BSA protein. It is a standardized protein solution used to create calibration curve by providing known concentration for
First
comparison.
things first, we need to adjust our pipette to 1000 microliters, since 1000 microliters is equal to 1 milliliters. After that, we need to pipette 4 milliliters of distilled water. That means we need 4 times pipette of 1000 microliters into a 6 test different tubes. Then we need to put 1 ml of Bradford reagent into each tube. Then we need to put 50 microliters of BSA protein standard, 100 microliters of BSA protein standard, and 200 microliter BSA protein standard. And we need to prepare each duplicate. That's why we need 6 tubes. That means there are 250 microliters, 100 microliters, and 200 microliters of BSA protein standards. After the solution is mixed well, we need to pipette the solution, 2 ml of the solutions, into an empty cartridge to record an absorbance. In order to get absorbance from each test tubes that contain protein, we need a blank sample. Blank samples are coming from the test tubes that without proteins. And that is the end of the part 1. In the second part of the experiment, we need to find each absorbance of unknown protein A, unknown protein B, and unknown protein C. The part of the experiment is very similar to part 1, just replaced with unknown A and unknown B and unknown C. Like in experiment 1, we need to use the test tube without protein, act as a blank to find each observance. That's all of part in the experiment. And these are the results for experiment 1, part 1, and this is for experiment 1, part 2.
In conclusion, part 1 of the experiment successfully demonstrated the absorbance increase with the increasing protein concentration, which follow the Beers-Lambert law and allow the construction of reliable calibration curve. In part 2, the calibration curve was successfully used to determine the concentration of the unknown protein sample based on their absorbance value. Among the sample tests, sample C showed the highest protein concentration, followed by sample B and A. Overall, the experiment confirmed that spectrometry is an effective method for quantitative protein analysis. From this experiment, we learned how to use spectrometer to measure absorbance and understand the relationship between the absorbance and the protein concentration using the Beer's-Lambert's law. We also improved our skill in preparing standard solution, performing accurate pipette, and analyzing data using the calibration curve to determine the unknown concentration.
As challenges for our experiments are consistent of the results. Our results in the first run are some of them high, some of them are low, which is very inconsistent and not following Beer's-Lambert's law. As we know, Beer's-Lambert's law, concentration increase, the absorbance is increased. And as a second run for our experiments, we got the result we need.
Some of the challenges that are faced during this experiment is the workspace constraint, where 5 of us have to work on a small bench area, and the other is dividing the labor because some of us have to do the experiment and some of us has to record the experiment. This is hard to ensure everyone has a hands-on experience of using every machine that is included in the experiment.
For suggestion on improvements, I think all samples can be prepared in 3 replicates, and then we need to ensure that the temperature is stable, such as all the tubes are cooled to room temperature before reading,